For lab 3, we're going to do the cell cycle, which is mitosis. This is going to be the process of the cell developing, growing larger, and then finally reaching a point where it divides and produces daughter cells. Now your cells have their own life cycle, just like you have a life cycle. They start out small, they have to grow for a while and metabolize, then they get ready to divide, and then they will divide and produce 2 daughter cells. In mitosis, this is cloning, so they will be identical to the parent cell. Now, with the cell cycle, we start out with different names. The beginning of it is G1, which is the growth and metabolism of the cell from small to larger. Then when it's ready to divide, it will enter into the S phase. S for synthesis, and this is when it will double the amount of DNA so that it has enough to give each daughter cell. Then we'll go back into G2, where it's doing the final preparation, making sure all the proteins are produced, and then all the organelles are ready so that when they do divide, they not only have to give the daughter cells the DNA, but they have to separate the organelles so they get enough of each type of organelle so each daughter cell will survive. When they go through the process of dividing, it is called mitosis. This is actually the division of the DNA, and it's going to be sort of like a dance of the chromosomes. First, we're going to condense and find our partners that we've doubled already, and find our immediate partners that look exactly like us. And then we will hang out together with a little belt around our waist so we don't lose each other. Then we'll move to the middle of the cell and then be pulled back to opposite sides. This whole process takes about 20 minutes in the human body. Some cells it takes longer, some are shorter. Most of the cell's life are going to be during that interphase period, which is going to be comprised of the G1, S, and G2. Inter because it's between the divisions. So we have before us some models, and I'm going to hold up the different ones, and we're going to go over the different parts of the models. First, we're going to start out with interphase. This is interphase. During interphase, this is what the cell normally looks like. So at this point, we have the nucleus is intact. The DNA, which is this purple area here, is in the chromatin phase. It's stretched out and relaxed. We have a nucleolus still present, and we're going to look at these centrioles because these are going to be involved with cell division. They are located to one side of the nucleus, and at this point, the cell is just doing normal growth and metabolism. It will then enter into prophase. Now, we can break prophase down to early, late, middle. I'll show you quickly the changes as it goes about. What we first see happening in prophase are the centrioles are starting to move, and they're going to move to opposite poles eventually. And the DNA has condensed and found their identical partners. Remember, we reproduced our DNA in the S phase. So now there's one that looks exactly like me, and another one that looks exactly like me, or 2 of each of you in the class. And we find our partner and we put this little belt on, this little pink spot here. It's called the centromere in the middle of the chromosomes. These are called sister chromatids. They are identical sister chromatids. Okay? And notice the nucleolus is disappearing a little bit and the nucleus is going to break down. By the end of prophase, the cell will now look similar to this. The centrioles are at the opposite poles. The nucleolus and nucleus are completely disappeared. The DNA is in their partners, the sister chromatids. They are not organized at all, and coming from the centrioles is the mitotic spindle, which are these fibers that will go out towards the center of the cell. They're made up of microtubules. Okay. The next phase we're going into is metaphase. Meta, think of the middle. At this point, those sister chromatids line up all in a line on the metaphase plate in the middle of the cell, or equatorial plate. They are going to be attached to the muscle, the spindle fibers at a certain area on the centromere, and eventually they will release each other and move to the opposite poles. The next phase is going to be anaphase. Anaphase again can be broken up into early and late, and we're going to start to move away from each other. The sister chromatids release each other. They start being pulled back towards the opposite poles by the mitotic spindle fibers. and they are separating. This is late anaphase, where they are very far apart towards the poles. The final phase is going to be telophase. Telophase, we start to form a nucleus around each group of the sister chromatids, which are no longer considered sister chromatids. They're actually chromosomes at this point because they have their own centromere. When you share the centromere, you are considered a chromatid. When you have your own, you are considered a chromosome. So we have groups of chromosomes at opposite poles. The nucleus is reforming. So we're forming two nuclei. Okay. At the end here, we have the two nuclei are intact. The cells are going to go back to normal. The nucleolus is starting to form. And we have cytokinesis occurring, which is the pinching in of the cell membrane, which is caused by a cleavage furrow, and it will pull in and eventually separate the two cells from each other, and you will no longer see that they're now back, that they're identical. And they're going to go back now to being small cells, and they're going to enter into interphase. In the G1 phase, they're going to start to grow until they are ready to divide, and then they will go into the S phase.